An illustrative synopsis of influenza virus nomenclature. Pause text and illustrations for detailed study. Refer to the previously posted constituent factors observed in viral diseases, parts 1 through 5, virus spike protein structure and life cycle illustrations, and antiviral properties of zinc for data relevant to this presentation. Definition of terms. Influenza, the flu, is an acute viral respiratory tract infection caused by influenza virus genera A, B, and C, observed under isolated, epidemic, or pandemic circumstances in human beings. Hemagglutinin, HA, is a viral capsid protein functioning as an attachment factor and membrane fusion protein. The matrix 2 M2 proton selective viroporin of the influenza viral envelope plays an essential role in proton ion conduction, resulting in viral replication and proliferation for infection of other host cells. Neuraminidase, NA, is a glycoprotein enzyme functioning in receptor cleavage, resulting in viral release as observed during influenza A, which mediates binding to cell surface sialic acid receptors in the presence of the hemagglutinin glycoprotein. HA and NA surface proteins perform as viral surface antigens, recognizable by the immune system and capable of triggering an immune response. Ribonucleoprotein, RNP, is a complex of protein and ribonucleic acid, RNA. Ribonucleic acid consists of the genetic material of RNA viruses and functions in the flow of genetic information in all cells. Influenza virus attaching cell membrane via the surface protein hemagglutinin illustration. Structure of the influenza virus glycoprotein neuraminidase illustration. Structure of swine influenza virus, showing neuraminidase marked as Na and hemagglutinin as Ha, and different antigen types on and inside the capsid. Note the matrix 2 or M2 proton channel in white. Influenza M2 protein closed state structure hydrogen ion channel illustration. This is a picture of an influenza flu A virus. Influenza A viruses are classified by subtypes based on the properties of the hemagglutinin or neuraminidase surface proteins. There are 18 different HA subtypes and 11 different NA subtypes. Subtypes are named by combining the H and N numbers. This illustration depicts a 3D computer-generated rendering of a whole influenza virus, rendered in semi-transparent blue atop a black background. The transparent area in the center of the image revealed the viral ribonucleoproteins RNPs inside. The RNPs are shown in white with their coiled structures and 3 bulbed polymerase complex on the ends. An influenza virus's RNP is composed of both RNA and protein. Every influenza virus has 8 RNP segments, corresponding to the virus's 8 total gene segments. 3 of these RNP segments encode the virus's surface proteins, i.e. the HA, NA, and M proteins. The virus's hemagglutinin and neuraminidase surface proteins are displayed in semi-transparent blue, sticking out of the surface of the virus. HA is a trimer composed of 3 subunits, while NA is a tetramer comprised of 4 subunits, with a head region resembling a 4-leaf clover. Understanding the naming of flu viruses. This image shows how influenza viruses are named. The name starts with the virus type, followed by the place the virus was isolated, followed by the virus strain number, often a sample identifier, the year isolated, and finally the virus subtype. The virus antigenic types are A, epidemic or pandemic, B, epidemic, C, generally mild non-epidemic, and D, affecting cattle. The host of origin can vary, being avian, human, equine, swine, etc. Hemagglutinin and neuraminidase antigen descriptions are notated in parentheses for influenza A viruses. No antigenic subtypes are recognized for influenza B viruses, although they do have distinguishable HA and NA lineages. Influenza C viruses are not classified into subtypes. Fujian influenza virus nomenclature illustration. Human seasonal influenza viruses. This graphic shows the 2 types of influenza viruses, A and B, that cause most human illness and that are responsible for flu seasons each year. Influenza A viruses are further classified into subtypes, while influenza B viruses are further classified into 2 lineages, B. yamagata and B. victoria. Both influenza A and B viruses can be further classified into clades and subclades, which are sometimes called groups and subgroups. Note that this graphic is an example, and currently circulating influenza clades and subclades may differ from those presented here. New presentations coming soon. Thanks for watching.